Alright, hello everybody, welcome back to another biohelp video. Today we're going to be talking about the cell cycle, mitosis, meiosis, and binary fission. First, we're going to get into mitosis. Mitosis is the process that's used by body cells or non-sexual cells called autosomes. This is a process that is quick and it replicates the cell to produce two identical cells at the end. The process can be displayed as a cycle, and there are three main phases that are distinguished as the parts of mitosis. So first we're going to start its interphase. First comes interphase, which is not a part of mitosis, but is a part of the cell cycle. Interphase consists of three parts, which involves growth of the cell, DNA replication, and more growth of the cell and the copying of organelles. This consists of the growth being G1, S phase, which is DNA replication, and the G2 phase. So first we're going to get in the interphase. The G1 part is the start, and it consists of growth and normal metabolic rates, meaning that is growing and keeping the constant metabolism going. And this is important as it preps the cell for the starting of the DNA replication. DNA replication, or S phase, means the synthesis of DNA, or replication. This is the part where the DNA is replicated and is doubled to be diploid, or double, of the chromosomes it normally has. This is so that when the cell splits, eventually, that the cell will be able to have all the DNA it has as well. G2 is the last part of interphase and includes the growing of the cell even more, and it doubles the organelles in the cell, so that when it splits again, that there's an even amount of DNA and organelles in each one. This marks the end of interphase, but is not the end of mitosis. Mitosis is just starting. Mitosis starts with prophase. Prophase starts in the beginning of mitosis, and this is the phase where DNA condenses into chromosomes, specifically sister chromosomes, which are sister chromosomes, which are attached at a centromere at the middle, where they kind of cross over with each other, make this X shape. Also, the nucleus, the nucleolus and the nucleus starts to dissipate, and the DNA will start to flow free in the cytoplasm. Next comes metaphase. Metaphase is the second phase, and this is where the chromosomes line up in the middle of the cell across a plane, called an equatorial plane, or a metaphase plate, or some other lines as well. And they line up on there, and then centrioles, these little tiny, the centrioles then produce spindle fibers, which then are these long strands, as you see in the diagram, on the ends of the cell, which will then, in anaphase, pull it apart. So anaphase is the third part, and this is where the DNA starts to split apart and move to opposite poles of the cell. This is preparing for it to split, as the DNA is now going to different sides and grouping together, so that when it splits, that there's going to be an even amount of DNA in each side. This happens as the chromosomes that are paired by the centromere, the sister chromosomes, will now go to opposite sides of the cell. So it breaks apart that centromere and then takes half of that chromosome with it. Now we're going to go to telophase. Telophase is the part of the cell cycle where the cell is now fully prepping to start division of the cell. The stage of where the nuclear membrane starts to reform, the DNA starts to change back into chromatin, condenses, and then it is also where the cytoplasm starts to prep for the final phase, which is cytokinesis. Cytokinesis is the last part of mitosis, and is the part where the cell splits into two separate cells, and the cytoplasm fully divides, and there are two cells. Now, at the end process, here's some characteristics of the cells. They are called daughter cells. The two daughter cells that come from mitosis are genetically identical. The DNA that was used and replicated is the exact same in each cell, so they're genetically identical. Another thing is they are both called diploid, or containing the double amount of chromosomes in the cell. Now, this is due to the replication of those cells, or the DNA in the beginning, meaning that when it splits, it's going to have a full set of chromosomes. Now, this is all for mitosis. Now we're going to start meiosis. Meiosis is just like mitosis, however, there's a few more phases than there is there, and a little bit more complicated. So, another thing is that meiosis is sexual reproduction and produces a gamete, or a sex cell. So, I have this list here on the right. This will kind of go through it. At first, we're going to go into interphase, just like mitosis. However, just like mitosis, interphase is needed again, where the cell will grow, it'll duplicate the DNA and the organelles and everything for this. But this is not the first time. So we're going to go. So meiosis occurs twice, basically, and we'll get into that a little bit later. But meiosis 1, prophase. This is the part in meiosis where the DNA condenses, the nuclear membrane dissipates, the nucleus goes away, the DNA turns into chromosomes, but they condense into sister chromosomes that form bivalents. Then those bivalents kind of touch and cross with each other and cross over some of that DNA to other parts. So now that DNA is different. It's not genetically the same. This is where meiosis is different than mitosis, as the genes cross over, one from the mom, one from the dad. You go swap that DNA. Alright, now we're going to go to metaphase. This is where the sister chromosomes start to align across the equatorial plane or metaphase plate, and the centrioles go to the end, so just like mitosis. Anaphase, the chromosomes go to opposite sides of the cell, they split at the centromere. And then we got telophase, where the nuclear membrane starts to reappear, DNA condenses, and the cell prepares to split. And then we have cytokinesis 1, where they split. I forgot to include the slide. However, cytokinesis 1, they split together and form those two cells. However, interphase does not happen again in meiosis. But it does go through that cycle again. But it doesn't grow, it doesn't duplicate the DNA. It's going to stay the same. So now those two cells are each going to do meiosis 2. Prophase 2 is going to be, it's just like prophase 1 of mitosis, where the nucleus starts to dissipate, the DNA condenses into sister chromosomes and are bound by centromeres, and the centrioles will also start to head to opposite sides of the cell. Metaphase 2, those two cells are then going to go line up in the middle, and then anaphase, they're going to split apart, opposite sides with the centrioles. In telophase 2 is where the cell prepares to divide the cytoplasm and make two cells, but that's for each of them. So 4 cells are going to be produced from this. So the nucleus comes back, and then the DNA uncondenses, goes back to chromatin, and then cytokinesis 2 happens. This is division of the cell to make 4 cells at the end. Right? Not 2, 4. So meiosis results into 4 haploid cells, meaning they have half the number of DNA from the original amount. Because if we see, we doubled it at the beginning, then we split the cells, so now they're back to the original amount, but then we didn't replicate DNA again. And now, since we split them again, each are going to contain half of the DNA originally. However, this does not create genetically identical cells. It's called genetic variation, and this why we need genetic variation is because some organisms reproduce and create clones of themselves, so they're genetically identical. But if a disease were to come through, they're not able to evolve or adapt to have that resistance to the disease, and they'll all get wiped out. That's why genetic variation is needed. It's also to adapt and to evolve as well. Because since you're not getting a genetic variation crossing over between the mom and the dad, everyone's different. Everyone's got different genes in that, so it would allow for genetic variation. Next, we're going to go to binary fission. So, like mitosis, but instead it's done by prokaryotes. So I have a little diagram on the right. This process makes 2 identical prokaryotes at the end of the process. They're going to, replicate their DNA. They're going to grow. They're going to duplicate their structures, which are called plasmids. They're ribosomes. Plasmids are just little tiny bubbles they have with DNA in it. And when they grow, they're just going to split that long uncondensed DNA to different sides of the cell and just go right down the middle. And they're going to make 2 cells that are identical to one another. And this wraps up today's video. Thank you for watching. Stay tuned for the next video of translation and transcription, where we talk about how the DNA is replicated and how proteins are made. Thank you.